Proteins are constructed from amino acids. One of them, alanine, is shown here. An amino acid includes a positively charged amino group, a negatively charged carboxylate group, and a side chain. Each of the 20 amino acids used to build proteins has a different side chain. Two amino acids, for example alanine and threonine, can undergo a condensation reaction. The carboxylate group of alanine reacts with the amino group of threonine. Two hydrogen atoms and an oxygen atom form a water molecule in a condensation reaction. The amino acids are now linked by a covalent bond, an amide bond called a peptide bond, and the molecule is known as a dipeptide. The remaining structure of each amino acid is called a residue. A third amino acid can be added to produce a tripeptide. Additional amino acids produce a longer chain. In cells, the mechanism of polypeptide synthesis is more complicated than what is shown here. This polypeptide chain contains 6 amino acid residues. The amino and carboxylate groups of the original amino acids lost their positive and negative charges when they formed peptide bonds. But the amino group of the first residue and the carboxylate group of the last residue remain. These groups mark the two ends of the chain, known as the amino terminus or N-terminus, and the carboxy terminus or C-terminus. Notice that the side chains of the amino acids do not participate in forming peptide bonds. Most of the polypeptide chains made in cells are much longer than the ones shown here. A protein molecule may contain one or several polypeptide chains. In theory, a polypeptide could contain any amino acids in any order. Since there are 20 different amino acids, an astronomical number of amino acid sequences is possible. In a cell, polypeptides are constructed by ribosomes, which add amino acids in the order specified by a gene. This is known as a protein's primary structure.